Hello friends, today we are going to discuss about anti-pseudomonal drug. So this was the question which was asked in June 2022 DRNB critical care exam, and the question was enumerate group of antimicrobial and their mechanism of action. So the anti-pseudomonal drug classification is beta-lactam antibiotic, aminoglycoside, polypeptide, and quinolones. Among beta-lactam, the penicillin group which are active against pseudomonas is carboxypenicillin and the uridopenicillin. Carboxypenicillin include carbenicillin, ticarcillin. Uridopenicillin include azlocillin, meslocillin, piperacillin. Among Monobacter, Astronam, cephalosporin, ceftazidime, cefoperazone, cefepime, and cefepirone. And among carbapenem, imipenem, meropenem, doripenem. Aminoglycoside group which are active against pseudomonas is tobramycin, amikacin, gentamicin. Among polypeptide, poly B, polystine. And among quinolones, ciprofloxacin, levofloxacin, pfloxacin. Vancomycin is not effective against Pseudomonas. So mechanism of action, beta-lactam ring is the key to mode of action of this drug that target and inhibit cell wall synthesis by binding enzyme involved in the synthesis. These enzymes are anchored into the cell membrane and as a group is referred to as a penicillin binding protein. So, bacterial species may contain different contain between 4 to 6 different types of penicillin binding protein, and these penicillin binding proteins involved in cell wall cross linkage and are often more critical for survival. So, four-membrane ring of the beta-lactam antibiotic gives this compound three-dimensional shape that mimics DALA-DLA peptide terminals that served as natural substrate for transpeptidase activity during cell wall polypeptidoglycan synthesis. Tight binding of this beta-lactam drug to transpeptidase active site inhibits cell wall synthesis. So death results from the osmotic instability caused by faulty cell wall synthesis or binding of beta-lactam to PBP may trigger series of events that lead to autolysis and death of the cell. Beta-lactam agents are active against gram-negative and gram-positive organisms, but effectiveness varies owing to the structural difference in cell wall structure and PBP content. So this is the diagrammatic representation of mechanism of action of beta-lactamase. So next class of action is next class of drug is aminoglycoside. Aminoglycoside have the bactericidal activity in which they bind to the bacterial ribosome 30S subunits. Specifically, they are believed to bind A site on the 16S RNA component of ribosomal 30S subunit. Through this binding, genetic code get misread and the translocation is disrupted, leading to bacterial being unable to carry out protein synthesis. So the next class of drug is polypeptide. The cationic antimicrobial peptide exert antibacterial activity by interacting with negatively charged bacterial membrane to increase membrane permeability, lead to cell membrane lysis and cell content release. Upon approaching the cytoplasmic membrane through electrostatic interaction with microbial membrane, AMP bind to microbial membrane and interact with the anionic component of the plasma. Prior to this, antimicrobial peptide have to pass through capsular polysaccharide and other component of cell wall, such as liposomal polysaccharide of gram-negative bacteria, lipoticoic acid, peptidoglycan of gram-positive bacteria. Studies have shown that alpha helical AMP bind to the anionic lipid membrane and transform its disordered structure into aqueous solution into amphiphilic alpha helical structure to facilitate interaction with the membrane. Different forms of the alpha helical peptide, the beta sheet peptide, do not undergo major conformational transition when interacting with the membrane due to their stable disulfide bondage bridge. The peptide lipid ratio is the another major factor that affect AMP interaction with the cell. A low peptide lipid ratio AMP are located in parallel on the surface of plasma membrane. With increase of peptide lipid ratio, AMP are vertically oriented and inserted into hydrophobic center of the membrane. Eventually, membrane permeation lead to the leakage of intracellular membrane metabolites, biosynthesis, and the consequent cell. So this is the diagrammatic representation of mechanism of action of polypeptide. So the direct bactericidal mechanism is performed through interacting with the negatively charged membrane, resulting into increased membrane permeability, cell membrane lysis, and release of intracellular content, which ultimately leads to cell death. There are four main model of membrane pore formation, that is barrel stave model, toroidal pore model, carpet model, aggregate model. After AMPs penetrate into phospholipid membrane, their hydrophobic region combines with internal hydrophobic region of phospholipid bilayer, while their hydrophilic region are exposed to outside. Another bactericidal mechanism is AMP penetrate into cytoplasm, interact with the intracellular substrate, such as inhibiting RNA, DNA, protein synthesis, inhibiting protein folding, and inhibiting enzyme activity, cell wall synthesis, promoting the release of lysis to destroy cell structure. The next group is fluoroquinolone. Fluoroquinolone act by inhibiting two enzymes involved in the bacterial DNA synthesis, both of which are DNA topoisomerase that human cell lacks and that are essential for bacterial DNA replication, thereby enabling this agent to be both specific and bactericidal. DNA topoisomerase are responsible for separating strands of the duplex DNA, inserting another strands of the DNA through break, and then releasing originally separated strands. Thank you guys.